Welcome back, everyone. So today we're going to be talking about essentially three things: the cell cycle overall, what homologous chromosomes are, and what's up with sister chromatids. And I'm doing this primarily because I have students that I've noticed over the years get confused about these topics. They are confusing, and so what it all means, I need to do a quick screencast on that, and so that's what this is. So let's take a look what we mean by these different topics. So before we start talking about the cell cycle, I want to talk about what homologous chromosomes are. So if you look over here on the left-hand side, we're dealing with homologous chromosomes. Notice this is a chromosome. This blue one on the left and the red one on the right are actually chromosomes. They just are not drawn in the typical way that you think of when you think of chromosomes. The one on the left and the one on the right, they both only have what's called one chromatid. They've only got one part of the chromosome here. They don't have a duplicated or two chromatid structure to their chromosomes. They are still chromosomes. Both of these are still chromosomes. These are still chromosomes, and what is important also about these is that these are homologous chromosomes. What does that mean? Well, that means they code for the same traits. So an example of trait that they could be coding for would be this blue one over here, this red one over here represented by red and blue lines on these chromosomes. They would both code, for instance, of the trait of hair color with different alleles. That means different genetic options for that trait. So the general concept would be like a trait of hair color, the specific alleles for that, the specific genes that we're dealing with would be maybe dark hair from one parent and light hair from another parent. Those would make these two chromosomes homologous chromosomes. So that means they're going to be coding for the same things. That genetic information comes from different parents. All right, so let's take a look at other examples of homologous chromosomes. These are also chromosomes on the right. And so you've got this structure over here, this structure over here. Both of them are also coding for the same genes. And so the example that you see over here is a gene for dark hair on the chromosome on the left. Now notice that's been coded twice. So you've got a duplicate of that information. This chromosome on the left has identical sister chromatids here, as does the chromosome on the right. It has identical sister chromatids as well. So over here you could say you have a gene for dark hair. On the right you would say you have a gene for light hair, but they are both coding for the same things. The difference between the examples on the left and the examples on the right is that the examples on the right are going to have two chromatids for each chromosome. They're still homologous chromosomes. They're still coding for the same traits. It's that these traits have different alleles, like the gene for dark hair, the gene for light hair would be on another one, and the red duplicated chromosome would simply have two sets of that. And the blue duplicated chromosome would have two sets of that joined together. They're still called a chromosome. This one on the left over here and this on the right over here are simply one chromosome each. Now, I do want to point out that the red chromosome and the blue chromosome overall are homologous to each other, but they are not homologous to other chromosomes. So I've drawn another chromosome here. You can tell it's another chromosome because it has a different length. Usually chromosomes are separated by lengths. And notice, so this is a completely different chromosome. So that means this gene here would code for something completely different. Like, instead of the color of someone's hair, maybe it would be the shape of someone's thumb. Or maybe it would be whether or not they can roll their tongue. Just a completely different trait that we're talking about here. And so they are not the same style of chromosomes. So let's see how this plays out in the cell cycle. And so when we talk about mitosis, or when we talk about meiosis, we can also talk about the cell cycle. And this is the cell as it's going through its life cycle. It's not exactly the same as a human being being born, going through childhood, early adulthood, adulthood, older adult, and eventually dying. It's not the exact same idea as that, but it's similar enough that you can start to think about it in kind of those ways. So let's go ahead and start over here with a young cell. So with a young cell, we've got chromosomes, and these are homologous chromosomes that I've drawn here. These chromosomes are just one pair of homologous chromosomes for human beings. There would be 23 pairs of homologous chromosomes. For other species, there would be a different amount. So 23 pairs of homologous chromosomes would give us 46 chromosomes in entirety. Now these chromosomes, if you look, they are in the state where they only have one chromatid per chromosome. And if the cell stops dividing and stops growing, if it's not in an area that grows and divides very often, it can just go into the G0 phase. It can start to go through just an interphase part of its life where it just does its thing. So if it's a nerve cell, it's going to fire off at appropriate times. If it's a muscle cell, it'll contract at appropriate times, and so on. And so that's entirely possible. So this is like an option right here where this cell could get out of the entire life cycle and stop growing and dividing and just grow and then remain in its current state for years even, especially if it's a nerve cell. If it doesn't though, if it's continuing to grow and divide, then it continues around here through the G1 phase. During this G1 phase, you're going to have parts of the cell besides the chromosomes are duplicated. And so all of that cellular machinery that's important there is going to continue. Then we get to the next phase called the S phase. And I want you to notice right here, we go from homologous chromosomes that have one chromatid each to homologous chromosomes that have two chromatids each. That's during the S phase right here. And so that's where DNA is being replicated, we say, or sometimes we say it's being duplicated. Then the cell is going to double check what's going on. So during the G2 phase, there is a double checking of the duplicated chromosomes for error that's going on, and the cell is getting ready for mitosis. The mitosis entails a topic of its own, so I'm just going to quickly say that you have PMAT here, prophase, metaphase, anaphase, and telophase. At the end of mitosis, you're left with cytokinesis, which is the pinching off of the cell. At that point, you've got two daughter cells that are traveling off, and the process could sort of repeat itself if it continues down below. If it heads off to the right, you could switch your focus over to this cell over here if you wanted to. It would do its own thing. But notice as we go through mitosis, you're going from chromosomes with two chromatids each, two chromatids for each chromosome, to cells with one chromatid for each chromosome. And that is important because we end up with two different cells. And so in the big picture of mitosis, you've got one cell here going into two cells, and that's what's happening over here and over here. So I hope this has been helpful. There is a follow-up screencast that I've done on meiosis. There may be one that I do on mitosis later on, although that has been done before. But hopefully this now makes some more sense.